Hi everyone, welcome back to our Scientific Apologetics course. Over the next two lectures, we will be tackling the topic of the origin of life. Back in the first lecture, I talked briefly about the fine-tuning argument where we looked at the different constants necessary in order there to be a life-permitting universe. These initial conditions are required for the existence of life anywhere in the cosmos. And with the absence of these very specific fine-tuned conditions, there would not be galaxies, stars, or planets even through which life could exist. But even though there were these fine-tuned conditions, there's no guarantee that life is going to evolve. They're necessary for life to exist, but they're not sufficient. So what does it take for life to form? How did it arise? Could it have risen by chance or some other undirected mechanism? Or is there something more that has to be at work? And these are questions that concern the origin of life that we're going to examine in more detail through today and next week's sessions. Prior to the 20th century, scientists were asking this question: how did life arise from non-life? It was thought that living things had some sort of vital force that set them apart from non-life. Organic substances could only arise from organic precursors. But then in 1828, Frederick Wohler was investigating the formation of cyanide. He heated an inorganic compound, ammonium cyanide, and he thought that he would get cyanide. But instead he got a crystalline substance called urea, which is an organic compound found in living organisms. And it showed that organic compounds could be synthesized from inorganic. And in fact, there was not some immaterial life vital force. And if organic compounds could come from inorganic, why not life in the same way? At the same time, there was the advent of Darwin's theory of natural selection, and there was a plausible means by which organisms could now develop new structures and greater complexity by undirected material processes. So why not the same with chemicals themselves? With the right environmental conditions, the right mixture of ingredients, could simple organisms form? Seeing these mechanisms and similarity to life, it led a scientist named Alexander Oparin to propose a mechanism for the origin of life based on purely chemical processes. So here what we see is an atmosphere containing a mixture of different chemicals like methylene, dicarbon, water, hydrogen, carbon dioxide. These then react with ammonia in the presence of lightning and UV to form amino acids, which then start to link spontaneously to form polypeptides. These get incorporated into something called coacervates, fat molecules that clump together in a sphere and repel water. Using this, the molecules that interact with one another form a primitive metabolism and eventually form the first living cell. So this was the proposed mechanism, but was it viable? Was there evidence for this? And so in the 1950s at the University of Chicago, Stanley Miller and Harold Urey conducted their famous experiment. They took what was thought to be the primitive atmosphere with methane, ammonia, hydrogen, plus water. They added a high voltage charge to simulate lightning, and what they found was amino acids formed, glycine and alanine. And this was celebrated as proof of Oparin's hypothesis. William Day, a scientist, said, quote, "It was an experiment that broke the logjam. The simplicity of the experiment, the high yields of the products, specific biological compounds produced by the reaction were enough to know the first step in the origin of life was not a chance event, but was inevitable." But was it? If you think about Oparin's model, a crucial step in the development of life is the formation of proteins that are the drivers of life. It was through proteins that cellular machinery works, carrying, delivering essential molecules, catalyzing reactions. Initially, proteins were thought to have simple, regular structures. And so it made sense that if you could form a lot of different amino acids from inorganic compounds with enough interaction over time, you would get the necessary proteins for life. But things start to really complicate as molecular biologists made important discoveries about proteins. In 1951, Fred Sanger determined the sequence of insulin and showed that rather than being made up of simple, repetitive sequences, proteins were made up of non-repetitive amino acids in an irregular arrangement, like beads on a string. No specific law governs why one amino acid has to be next to another one. Later on, they were able to determine the structure of proteins, and rather than being linear or regularly shaped, proteins showed an amazing 3D complexity. Nobel Prize winner John Kedrow first determined the 3D structure of myoglobin, and it showed an extraordinarily intricate and irregular three-dimensional shape that arose from a twisting, turning, tangled chain of amino acids. Kedrow himself said that the big surprise was that it was so irregular, almost lacking the kind of irregularity that one would anticipate, and seeing that proteins themselves are actually quite complex. Not only that, but they start to realize that proteins exhibit another remarkable property. In addition to their complexities, proteins also exhibit something called specificity. Although they're built from simple chemical building blocks, their function as enzymes, signal transducers, structural components of the cells, that all depend specifically on the specific sequence of the amino acids. It's that sequence that gives rise to their 3D structure. The structure in turn determines their stability and also the function in which it can perform in a cell. And so in a functioning protein, its 3D shape gives it this famous 3D hand-in-glove fit with which it matches with other molecules to catalyze their reactions or build structures. And because of this fit, one protein can't substitute for another. The structure is very specific depending on for their function. And because the 3D function structure is determined by the sequence of amino acids, the amino acids themselves need to be in a very specific order. To change one would change the structure and therefore the function of the entire protein. And this need for a specific sequence in order to produce a specified function is something called specificity. And so scientists refined their understanding of proteins. They found that they showed the properties of complexity and specificity. And interestingly, these are also the same characteristics that are found in information. And so I want to take a small digression and talk about information. What exactly is information? For example, when an assistant dictates something in New York and then sends it to LA via fax, something arrives in Los Angeles. But the paper itself didn't originate in New York, only the information. Nothing physical, and yet something did arrive, something more than matter itself. So information can be defined as an arrangement or string of characters that accomplishes a particular outcome or communication function. For example, lines of computer code that cause a program to run, or sequence of English letters that make up a sentence. And when we think about information, there are two important aspects that we have to know. First of all, there's something called information carrying capacity. According to information theory, the amount of information something carries is inversely proportional to the probability of a particular event occurring. Okay, inversely proportional to the probability of a particular event occurring. So it's kind of a lot of jargon, but here's an example. So I live in Pittsburgh, where it rains all the time, especially in June. It's known as the second gloomiest city after Seattle. If someone told me it's going to rain in June, well, that's not very informative because it's a very high probability. But say a time-traveling weather forecaster came who's always right, and he tells me it's going to snow on June 20th. That would be informative, something not predicted. So the more improbable, the more information we get. So mathematically, if we look at a list of characters like this, what would make one more improbable than another? And two things that jump out right away are the number of possible characters per position, as well as the length of the character string. And so, for example, the probability of this top string would be 1 over 26, because 26 possibilities, letters per position, raised to the length, 16. That gives us a probability of 2.3 times 10 to the negative 23. The bottom's probability would only be 1 over 2, because it's only zeros or ones, to the power of 14, or 6 times 10 to the negative 5th. And we see that these two strings carry very different carrying capacity, even though they look kind of similar in length. The other aspect of information carrying capacity that's important is something called regularity. If it's a repeated pattern like below 0, 1, 0, 1, 0, 1, 0, 1, that's much less improbable than irregular random arrangement. And so this is what we mean when we talk about complexity. It's what Sanger observed when he saw an insulin molecule, an irregular arrangement of a wide range of possible amino acids extending 110 amino acids long. And it shows that this complex protein are things that are improbable and exhibit an irregular arrangement. But it's not just enough to have an improbable string of symbols. There's a second aspect to information, and that's called information content. And I'll illustrate it like this. Take these two strings of character, 4 and 7 years ago, versus these random strings of letters. You look at this, you instantly know one communicates something while the other one doesn't. Why? Well, in terms of complexity, they're actually equally complex. Each is made up of alphabet characters of equal length and without regularity. And therefore, they have a very similar probability, in fact, equivalent. But the chances that you randomly get the first is exactly the same chances that you would randomly get the second. So what tells them apart? Well, it's the specific arrangement of the letters that then communicates a function or meaning, in this case, part of the Gettysburg Address. And this same information content or specificity is what we see in proteins. That's why Francis Crick, he likened proteins to a linguistic text. And in a linguistic text, the meaning of the text depends on the specific sequential arrangement of the letters. In the same way that in proteins, the specific arrangement of amino acids give rise to a protein's function. And so what's intriguing about information is that we recognize information as originating from thought. Some sort of conscious or intelligent activity. For example, when we think about the SETI project right here, Search for Extraterrestrial Intelligence, when we're listening out there for electromagnetic signals, how is it that you can tell something's noise versus someone is really out there? Well, you look for these two things. Complexity. How long? How improbable is the signal? Is it an irregular arrangement or not? And then, does it communicate something? Is it specified? If the answer is yes, then if it's complex and it seems to communicate something, what do you think? Well, you think there must be some other life form out there. Because information seems to import intelligence. So what do we make sense of? What do we make of the information that we're seeing in these living organisms? Could these have arisen by artificial selection without some appeal to intelligence? Well, when looking at proteins and their specificity and complexity, what happened? It caused scientists to actually go hunting for the source. There must have been something that had caused all these things to form in these really complex proteins. And so when they went searching, that's what led them to the discovery of DNA. But before we get there, let's go ahead and do a quick review. So what did we go over so far? We saw that Frederick Roller's experiment to produce urea from inorganic compounds opened the door for Oparin's mechanism of the origin of life, purely based on chemical processes. And this viability of this process seemed to be confirmed with the Miller-Urey experiment, where they were able to produce amino acids from the elements of the primitive atmosphere. But the theory of life arising from undirected chemical processes became complicated with the further study of proteins, showing that proteins are both complex and specific in their sequence and structure. Their function is precisely determined by the sequence of amino acids and the resultant structure. And these are the very same characteristics that we find in information. And so then we went into some information theory. We looked at the concepts of information carrying capacity, which is a function of length, a function of number of possibilities per position, and irregularity. And then we looked at something called information content, which is the specific pattern that communicates something. And as we looked at those pieces of information, we considered that the patterns of information being complex and specific, well, these are typically linked with some sort of intelligence, just like in the SETI project. All right, so going back to our story. So back to the search for the source of specificity, complexity of proteins. Well, when they searched, that led scientists to the discovery of DNA, the source of the complexity they were seeing in the proteins. In 1953, Watson and Crick, they elucidated the structure of DNA, and soon after they found that DNA stores all the information necessary to direct protein synthesis. Further studies showed that the arrangement and sequences of these 4 nucleotide bases determines the sequence of all the amino acids that then determines all the proteins folding and structure, right, through transcription and translation. And how this all works is through something called a genetic code. Because it's like one of those things you'd find in a spy novel. And what the body does, it converts these bases, triplets of bases, 3 bases at a time, to a specific amino acid. Change one letter of the DNA, and it changes the amino acid, and it can change the protein itself. In other words, the specificity and complexity that we saw in proteins is encoded right there in the DNA. And as scientists peered into the cell, they started to discover a pretty amazing process that takes the material in the genome and produces a functional protein. It's a process that goes from something called transcription to translation. I just want to take some time to appreciate what it takes to make a protein. And so we're going to go ahead and watch a video about it. Let's watch it now.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a 2-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function.
Just to emphasize how intricate this process is, as well as the interplay of DNA and proteins, I'm going to kind of nerd out a little bit and talk about more biology. So the first process is called transcription, and the main protein in this is called RNA polymerase. And what it does is it recognizes the start of the gene, it then unwinds the DNA, then it grabs complementary RNA bases, and then links them together to form something called a messenger RNA. There are all these other proteins that are involved that I don't show that inhibit or promote transcription or even chop up the mRNA to edit it. After that is a process called translation, translating that code into proteins. And so what happens there? There's this big thing called a ribosome. A ribosome is 50 proteins and 3 long RNA molecules and 2 subunits. And what it does, it facilitates the pairing of the transfer RNA to the messenger RNA. And then what it does is then takes the amino acids that are carried along and starts to form them into a polypeptide chain that will then become the protein itself, just like we saw in the video. What's further complex is this transfer RNA piece, right? It brings the amino acid to the ribosome. There are 20 different ones of these, each a specific shape, specific for their triplet codon as well as the amino acid. And how do these things get loaded up? How do these transfer RNAs get loaded up? Well, there's this green thing called an aminoacyl tRNA synthetase. Long name, I know. But what's really cool about this is there's a specific one per tRNA, and it recognizes the transfer RNA as well as the amino acid and links them together in the right combo. And all these things together, that's what it takes to produce proteins from DNA. The combination of all these things working in concert with one another so that life can happen. And without this, life could not form. The entire process is analogized to something called the CAD/CAM process, computer-assisted design and manufacturing. It's used in manufacturing things like airplanes. And so what happens is a CAD program is used to design part of an airplane. Engineers produce the specifications, and that produces a 3D drawing. All this info is then stored in binary code. And once they finalize the code, it then gets translated into machine code. Machine code then goes to a machine arm, which then is able to make the parts of an airplane. And it's strikingly similar to the cell's information processing system we just talked about. From the DNA code with the instructions translates to genetic code that then tells the machinery how to form the proteins and starts to build out the life of a cell. But with even one more difference. The cell's information processing system not only does all these things, produces machines, but also reproduces itself via DNA replication. All these processes dependent on the very proteins that the DNA encodes. And so with the interconnectedness that we're seeing of proteins and DNA, both sources of information, came something called the DNA enigma. Where did all this information come from? And then which came first, proteins or DNA? Without proteins, you can't decode the information in the DNA and turn it into something functional. It's like having a piece of computer code without a compiler. It's useless. But then without DNA, how did these proteins form? Where did they get their instructions from? A compiler without code is useless. And Richard Lewontin framed the problem in this way. He said, no living molecule is self-producing. Only whole cells may contain all the necessary machinery for self-reproduction. Not only is DNA incapable of making copies of itself, aided or unaided, but it's incapable of making anything else. The proteins of the cell are made from other proteins, and without that protein-forming machinery, nothing can be made. And so how did all this come about? With all these discoveries, it forced scientists to redefine the question of the origin of life and now have to explain these 3 crucial features of life. Number 1, they had to explain the origin of the system for storing and encoding the digital information in the cell. DNA's capacity to store all this information, how the nucleotides form to store the DNA in this manner, and then the development of the genetic code. Number 2, the origin of the large amount of complex, specific information we're seeing in the DNA. Where did all this information come from in the first place? And then third, the origin of the integrated complexity of the cell's information processing system. The fact that proteins are dependent upon DNA and other proteins, and vice versa. DNA is dependent on these proteins. Which started first? How did it all form? And so to answer these questions, there have been 3 basic approaches to explain the informational properties of life. The first one is called chance. The chance hypothesis that things form based on random molecular interactions over time. The second theory is something called necessity, which is referencing law-like properties that sort of forced these things to form over time. A third approach combines the two, chance plus necessity, and then there's a fourth, one that's still very controversial in the science community. But if these three approaches do not give a probable mechanism for the informational properties of life, could design or intelligence be a legitimate explanation for these questions? Perhaps the explanation that best fits the evidence for the origin of life. And that's what we're going to tackle in the next part as we hit each of these approaches in detail. So before I jump into the next session, let's review what we've covered so far. First of all, we learned that complexity and specificity found in proteins is encoded in the DNA. And there's a complex interaction between DNA and proteins in order to produce the materials necessary for life through transcription and translation. That process being similar to the CAD/CAM system in engineering. And with all this, we came to find the DNA enigma. Where did all this information come from? Which came first, DNA or proteins? And it calls scientists to redefine the question of the origin of life so it could explain the creation of the system for storing information, the origin of that information, and then the genesis of the complex information processing system that we see in the cell. And finally, what we did is we walked through different approaches to help explain this. Chance, necessity, chance plus necessity, or intelligence. And so, the first option we're going to examine is the chance hypothesis, whether random molecular interactions could account for the appearance of proteins and DNA leading to life. But before we can examine whether chance can be a viable option for the origin of life, we have to talk about the idea of chance itself. So what is chance? What does it mean that something happens by chance? Let's take this example. So say a gambler went to Las Vegas and played a game of roulette, where there's a 1 in 38 chance of winning. He played, he bet on the 16th hole, and boom, the ball falls to the 16th hole that he bet. And he won, we would say, by chance. In other words, there's a finite set of possibilities and a definite probability that through randomness alone, an event will occur. And when it does, it's by chance. But what if you found that the ball always fell into the 16th hole? Or whatever hole the gambler bet on, 16, 32, 10, didn't matter, whatever hole he bet on, he won. At that point, would you continue to say that, well, that's simply, quote, by chance? Or would you eliminate that option and choose another, like call security on him? Right? And so there are ways in which you can look at a situation that appears at first to be by chance, yet later on be able to eliminate chance as an option. And how do we go about doing that? And there are three concepts we need to talk about: improbability, probabilistic resources, and pattern recognition. So talking about improbability, going back to the gambler example. Say he bets on the 16th hole 100 times in a row, and he wins all 100 times. The odds are 1 over 38 to the 100th power. What would you say? Could you say it's by chance? You'd be like, no way. You'd say, you cheater. Why? Because chance is no longer appropriate given the improbability of getting the same number 100 times. And so we can say we eliminate chance when a series of events happens that deviates too greatly from the expected statistical distribution. If you think about all the possibilities of the roulette on a bell curve, to win 100 times is what we call in the rejection region, where if an event falls, you can eliminate chance as an option. And we call this improbability. How improbable does something have to be before we can eliminate chance? Well, that depends on the improbability of the event and how many opportunities we have to generate that event. For example, with roulette again, the odds of getting the same number twice in a row is 1 over 38 to the second power, or 1 over 1,444. Not great odds, but actually it's more doable given the number of times you have to try it, as well as the amount of time in which you had to do it, if you were to visit again and again and again and again. And that describes something called probabilistic resources, which is defined as the time and the number of trials that are necessary to make an event probable. In other words, how many attempts do you have to make an improbable thing probable? If you think about carnival games, that's why they limit the number of times you get to try, so that winning remains improbable for you. And what's really interesting is that we as humans tend to overestimate the power of our probabilistic resources in overcoming long odds. And here's a scenario to kind of illustrate this. Say a criminal, he's arrested and he's told that he needs to serve 10 years in prison, or he can stay in prison until he can flip heads 100 times in a row. What should he do? What does your intuition tell you? You probably think, well, even if it takes 2 or 3 years to flip the coin 100 times heads, that's better than 10. And so if I'm really diligent with flipping, after all, what am I going to do in prison? I can get it. But the question is, how long would it really take to flip those 100 heads? It actually turns out to be really, really, really hard. Even 1 trillion years would not be enough. Why? Well, the odds of flipping heads 100 times in a row is 1 over 2 to the 100th power, right? So that's a 1 in 1.2 times 10 to the 30th chance of getting it. And so say this man flipped every 5 seconds for 8 hours a day, 6 days a week for 10 years. How many trials would he have? It would only be 1.7 times 10 to the 7th trials. If he did a trillion years, which is 10 to the 12th years, it would still only be 1.6 times 10 to the 19th power. Not even close to what would be needed for a reasonable chance of success. And so we'd say that this man does not have the necessary probabilistic resources. He should just choose the 10 years. All right, and then the third concept, if you think about it, right, this definition of improbability as a statistically rare event, well, actually, improbable events occur all the time. Say I flip a coin 100 times. It's really improbable to flip it 100 times heads every single time. But if you think about it, if I were to sit there and flip a coin 100 times and write out all the results of what I did, what's the chance that you would flip exactly the same as I did? Well, actually, it's the same probability, 1 over 2 to the 100th power. And yet, when you look at these two trials, they're very different. Why? One's clearly by chance, the other one is not. And what's the difference? If you're tracking, this might sound familiar. It goes back to the idea of information. When we're talking about information content, specificity, it's the example of the random letters versus the Gettysburg Address. They're equally improbable, and one is clearly by chance, the other is not. And so what, in addition to improbability, excludes chance? What's the kind of outcome? Some sort of pattern that's significant. In the coin flipper's example, it's the fact that he gets head every single time. So pattern recognition, too, is a way to eliminate chance. Both improbability and pattern recognition in concert work to eliminate the possibility of chance. And so using these concepts of improbability and pattern recognition, we can then evaluate the chance hypothesis for the origin of biological information. What scientists hypothesize is this, that there's a prebiotic environment in which there are all these different elements and chemicals. These interact with each other in a series of chance collisions to form the elementary building blocks of amino acids, nucleotides. These then continue to interact and through a series of fortuitous collisions produce the first functional proteins, nucleic acids, and then eventually into cells. And on the surface, it seems plausible. You have all the pieces necessary in a certain area. Give it enough time. Give it enough interaction. You should be able to produce the building blocks and eventually the proteins and DNA. But can chance alone explain this? The first thing we notice in proteins and DNA is that they show a functionally significant pattern. Like we talked about before, the way that they're arranged means something, just like words. For example, you look at proteins, they're not just a random sequence of amino acids. There's a pattern that has to be maintained, or else they lose their shape and their function. You can't just have a random collection of amino acids that just works. No, in fact, what scientists have found over time is that a random arrangement of bases and amino acids, actually most of them perform no function at all. It'd be like taking a bunch of Scrabble characters and having people pull out letters and then write them on a chalkboard. Most of the combinations would be gibberish. It'd be really, really hard to get a word, let alone a sentence that conveys information, similar to the formation of proteins. And so the question then becomes, how improbable is it to get a functional protein out of amino acids? Even a short 150 amino acid protein. And so here are some calculations. First, to form a protein, you need to form something called a peptide bond. And when amino acids are put in test tubes, they form two kinds of bonds, peptide and non-peptide bonds with equal probability, or about 1 over 2. So for a 150 amino acid chain to form, the probability that it will form the right peptide bond is 1 over 2 to the 150th power, right? The length is 150. That gives us 1 in 10 to the 45th power. Secondly, for every amino acid, there are two forms, an L form and a D form. These are called optical isomers. Functional proteins tolerate only the L form, and yet amino acids form both randomly at equal frequency, 1 over 2. So to get the L form is 1 over 2 chance, multiply the 150th power also gives you 10 to the 45th power. And then finally, what's the probability that a protein of this length would actually be functional? Well, if you're able to get the peptide bonds and the right isomers to give you 150 amino acids, would it be functional or would it not? In 2004, Douglas Axe, a professor, investigated this, and he found that the necessary probability to form a functional protein is 1 in 10 to the 74th power. And so then the odds of forming a 150 amino acid functional protein is just multiplying all these different probabilities, and what you get is the total probability is 1 in 10 to the 164th power. What does that mean? Well, just to give you a perspective, there are 10 to the 80th power protons, electrons, and neutrons in the entire universe. So the odds of finding one at random is 1 in 10 to the 80th. The odds of a functional protein are much, much harder than that. And so already that sounds pretty impossible, significantly harder than a needle in a haystack. But perhaps, with a lot of time, a lot of chances you could get it. Because it seems like you can. But remember, the prisoner example we talked about earlier, with the coin flips and how we tend to overestimate our probabilistic resources to overcome long odds. And well, we can actually calculate the probabilistic resources in our universe and see if it's probable to form a functional protein just by chance. Remember, when we look at the probabilistic resource of the entire universe, it's made up of two things: the amount of time you have, as well as the number of interactions you get. And here's how we can calculate that. Well, how much time have we had to try this combination? Well, we have had about 10 to the 16th seconds since the Big Bang. And then how many interactions are there per unit time? Well, to figure that out, you can multiply number one, the number of elementary particles there are in the universe, times how fast that they can interact. Scientists have done all these calculations. There are 10 to the 80 elementary particles, and these elementary particles can interact maximum at 10 to the 43 interactions a second. And so, taking these three pieces of information, you can multiply them all together, and you get the probabilistic resources of the universe is 10 to the 139th possible interactions since the beginning of time. Compare that to the probability of getting one functional 150 amino acid protein by chance, 10 to the 164th power, and it's not even close. Not to mention that life didn't have the entire universe to form. The age of the Earth is probably only about 4.5 billion years old compared to the universe, 14 billion. And the probability is so low that it makes sense to reject the chance hypothesis rather than to accept it. And then think about this. That was only for one protein. Most proteins are actually longer than 150 amino acids. RNA polymerase is 3,000 amino acids. And then secondly, minimally complex cells, they actually require many proteins, not just one. The smallest organism found requires at least 250 proteins to survive. So the probability is even greater to get life. Another thing to add to this improbability is environmental factors. The Miller-Urey experiment assumed a mixture of methane, ammonia, and hydrogen with no oxygen in order to get their amino acids. That's not what we've been finding in the initial environment. In fact, we find that it contains mostly carbon dioxide, nitrogen, water, and free oxygen, where amino acids would not have formed as readily. Also, the byproducts of that interaction, that experiment, would have reacted with the amino acids and other molecules to actually inactivate them and form a sludge, rather than pairing them up to form proteins. And that's why, in that experiment, these byproducts had to be removed. And so, it is possible that this could have happened simply by chance. You can always imagine it, but like the prisoner example, flipping the heads 100 times, the odds are infinitesimally small, even more improbable than the entire probabilistic resources present in our universe. P.T. Mora, a senior researcher at NIH, he put it this way. He said, when in statistical processes, the probability is so low that for practical purposes, infinite time must elapse for the occurrence of an event, statistical explanation or chance is not helpful. And so in light of that, most origin of life researchers have decided to consider other theories that don't rely so heavily upon chance. Since the odds of a purely random process producing life is so small, scientists have concluded that some non-random self-organizing process must have been at work to help overcome these long odds. Maybe the nature of the molecules themselves or the environment favored the formation of proteins and DNA. And that's actually where we'll start next time. So briefly, this is what we went over in this section. We discussed first how to eliminate chance using these three concepts: improbability, probabilistic resources, and pattern recognition. We then applied these to the chance hypothesis, to which proteins and DNA arose out of prebiotic conditions through a series of chance collisions. And we found that the probability of a single functional 150 amino acid protein arising by chance is significantly greater than the probabilistic resources of the entire universe. Not only that, but proteins are generally longer than 150 amino acids. The minimally complex cell requires over 250 proteins for life, and the environmental factors would have hindered the formation of these proteins. And all these together make it pretty much impossible to explain the origin of life simply by chance alone. Hi everyone, welcome back. Today we'll be jumping into part 2 of our section on the origin of life. Said a lot last time, so I want to do a quick review. Last week we talked about the origin of life question and the theory that life arose out of inorganic molecules by an undirected mechanism. At the time, it seemed plausible to scientists because of the picture of what proteins were, and of the cell, they were actually quite simple. But as they started to uncover the intricacies of proteins and the underlying genetic code found in DNA, they discovered that what was contained was actually a lot of information. we saw that the sequence of bases and amino acids had the qualities of being complex and specified. Not only that, but the interconnectedness of DNA and proteins led to the function required for life pose a puzzle to origin of life researchers. How did all this arise? And it forced scientists to redefine the question of the origin of life to explain three crucial factors of life. Number one, the origin of the system for storing and encoding information in the cell. Number two, the origin for the large amount of complex and specific information found in DNA. And number three, the origin of the integrated complexity in the cell's information processing system that was required for life: transcription, translation, and replication. We looked at the chance hypothesis, looked at the ways that we can eliminate chance by the concepts of improbability, probabilistic resources, and pattern recognition. And then we applied this to proteins, and we calculated the possibility that chance alone could form a 150 amino acid functional protein. We found that that probability exceeds that of the entire probabilistic resources of the universe. That's just for one protein, let alone the more than 250 proteins required for life, as well as the further discovery that the early Earth environment is actually not suitable for the formation of these molecules. And so when we put these things together, we saw that the possibility of life arising by chance alone is extremely low. And so what are the other options? And here we want to turn to necessity. And so Christian Dudouvet, a Nobel Prize-winning biochemist, said, what is necessity? It's a string of improbable events drawing the same lottery number twice or the same bridge hand twice in a row. That doesn't happen naturally. All of which leads me to conclude that life is an obligatory manifestation of matter, bound to arise when conditions are appropriate. And what he's talking about here is another explanation called necessity, saying that there must be something intrinsic to the chemicals and molecules that causes them to assemble in a way that creates life, kind of like a snowflake. And it's really intricate, it's complex, it almost seems created, but it's not. It forms as a product of water vapor that crystallizes into a six-sided crystal because of the structure of water and then the slight changes in the surrounding humidity and temperature that causes a variation in the arms. All this is non-random, directed simply by the laws of physics and chemistry. And so this idea of necessity leading to the origin of biological information was investigated at length by someone named Dean Kenyon at San Francisco State in the 70s and 80s. He and another colleague, they titled this process biological predestination in their seminal work. And in their book, they presented their new theory on the origin of biological information by talking about something called self-organization. In physics, self-organization refers to a spontaneous increase in the order of a system due to a natural process, a force, or a law. And so here's an illustration. When a bathtub drains, you see a vortex forming. This vortex is a product of gravity acting on the draining water through a small hole, and it generates this complex structure, and it's increasing the order of the system. And so they tried to explain the origin of life by attributing the organization and complexity that we see in living things to physical and chemical forces that could be described simply using the laws of nature. And Kenyon, he then set out to describe these law-like processes that could generate life and the critical components of the cell from some sort of initial conditions. In other words, he's trying to figure out a way to overcome the long odds of chance itself in producing life. Kenyon's model is actually very similar to Oparin's regarding the progression of increasingly complex molecules, except that rather than chance driving these transformations, he relied exclusively on directed chemical processes. So for example, when Oparin said that monomers became polymers, like these amino acids became polypeptides, simply through random interactions, Kenyon said no, it's actually through forces of chemical necessity that are bringing these things together. He pointed to something like salt and how the electrostatic forces bring sodium and chloride together and it forms this highly ordered crystal. He says similarly, when you look at proteins and amino acids, there must be some sort of special affinity, attraction to one another that causes them to arrange in the form of proteins and life. And he first came to this idea when doing different experiments called dimer experiments. He wanted to see whether certain amino acids bond more readily with other amino acids. And so he tested all these different combinations of amino acids, and they found that they actually do bind differentially. For example, glycine forms linkages with alanine twice as frequently and twice as readily as glycine with valine. They also found that there are differences in attraction between amino acids depending on the differences in their structure. For example, those with longer amino acid side chains bonded less frequently than those with one another than those with shorter side chains. And looking at that, if that's the case, then there are these properties of these individual amino acids could themselves help to sequence them into chains and give rise to functional proteins. You know, it's kind of like these magnet tiles that we play with as kids that snap together with magnets into their shape. The sequences themselves that would have been completely improbable by chance alone can now be probable when these affinities are taken into account. And he hypothesized that proteins through these mechanisms arose first, and then later DNA and RNA arose. But then, could this self-organizational model really explain the information in DNA? If Kenyon was going to hold to this self-organization, this necessity approach, then there are really only two explanations for the appearance of DNA. Either, number one, specific sequences of amino acids and proteins provide the template for sequencing the newly formed DNA, or DNA itself self-organized in the same ways that proteins do. But as you think about these two different hypotheses, there are issues with both of them. First of all, with amino acids as a template. When you look at biochemistry and how the cell works, you find that always information flows from DNA to the proteins. It's not the reverse. You don't see proteins serving as templates to make DNA. And there's good reason for that. First of all, in the genetic code, each triplet of DNA specifies exactly one amino acid. So let's look at the genetic code again. Let's look at this sequence of UCU in the upper row. It encodes for serine and no other amino acid. And yet the converse is not true. Most amino acids correspond to more than one triplet, like serine to these four triplets. And so the flow of DNA to protein ensures that information flows without loss of specificity, and it's not true in the other direction. Secondly, proteins make really poor candidates for replicators. Replicators are molecules that function easily as templates for copying. Unlike DNA, they don't have anti-parallel strands of identical information, and therefore they can't be copied in the same way. They can't hold their fidelity. Proteins also are highly reactive once they become unwound or denatured. That's why most proteins tend to cross-link and aggregate or become destroyed when they're denatured. For example, when you look at eggs, when they're heated up, they start to denature the proteins, becomes opaque. These proteins aggregate into a solid mass that is then irreversible. So you lose stability and you lose function. That's not good to replicate. And third, new findings show that although amino acids have different binding affinities with one another, these actually don't correlate with the sequences that we find in known functional proteins. For example, look at glycine. With glycine forms these different bonds. If it bonds with alanine twice as better than with valine, then you expect that there'd be 2 times as much of glycine-alanine bonds than the glycine-valine bond. That's not the case. And so these differential affinities, though you see it in the test tube, they don't explain the specific sequences of amino acids found in functional proteins out in life. And so all these findings point to the fact that the sequences of the proteins did not form DNA. But in fact, the reverse is true, that the information encoded in DNA determined the sequence of the proteins themselves. So then the second question, did the DNA self-organize to form the DNA molecule? But this too had some issues. The first issue was actually more philosophical. When you think about the nature of information, information does get conveyed along mechanisms that are governed by the laws of nature. For example, fax machine, the circuits that store the binary code, the telephone wires that transmit across the country, the inkjet that prints it all out, all of these are governed by physics and chemistry, by law. But these laws do not govern the higher level properties of information itself, the words, the sequence of the text, the ideas. None of that is governed by law. Just as the properties of acoustics and air, they don't govern which sounds are produced by a person. And similarly, when you look at DNA, while the structure, the helix, the binding between the bases, they're all governed by chemical law. The specific base sequences that encodes the information for proteins, these are not. The genetic code must be explained by something else. Also, DNA's ability to convey information really requires freedom from chemical restraint. If the bonding of nucleotides would determine their arrangement, then the ability of DNA to convey information would be destroyed. Like this example in the alphabet. If we said that most times after a B, there had to be an A, and whenever there's an A, a C or a D has to follow, right, how many words could you actually make? Back, bad, not many. Pretty soon a rigid pattern would emerge and it'd be impossible to store much information. Similar for the DNA. Michael Polanyi, he wrote about this. He said, whatever may be the origin of a DNA configuration, it can function as a code only if its order is not due to the forces of potential energy. In other words, self-organizational laws or necessity. It must be as physically indeterminate as a sequence of words is on a printed page. The second issue that has to do with this has to do with the chemistry of DNA itself. Even though philosophically it doesn't make sense for DNA to have determined affinities, is that really actually true in real life? Could it be that certain nucleotides preferentially bind to one another, just like we saw with amino acids, and open up the possibility of self-organization? Well, to get at this, we have to zoom in on the structure of DNA. So what is DNA? DNA is a double helix consisting of repeating motifs. And we examine the structure of DNA, here are where the bonds are. There are bonds between sugar and phosphate molecules, forming the twisted backbone of DNA. There are bonds between individual nucleotide bases and the sugar-phosphate backbone. And then there are hydrogen bonds between individual nucleotides of opposing strands. But what's really interesting is where there are no bonds. There are no chemical bonds between the bases along the longitudinal axis in the center of the helix. And yet this is exactly where information is stored. The sequence of A to G to C to T. It's precisely on this axis that we're talking about when we're looking for differential affinities, where the one base binds more preferential to another base or not. And here we find no bonds at all. But perhaps the affinity is not at the base level. Could it be at a different site along the DNA that causes it to bind differentially to one another? But if you look at the structure of DNA, what do you find? The place where the different bases bind to one another along the backbone, well, it's exactly the same bond everywhere. And the N-glycosidic bond. Doesn't matter which base is attached. Doesn't matter where it is. None of them is more favored than another. And so in sum, when you look at the structure of DNA, there are no bonds on the information axis between the A, G, T, and C. And there's no differential binding even on the backbone and all other places where the bonds are. And so the analogy would be something like this. The analogy of magnetic letters. So here you have a force between the letter and the surface. That's similar to the N-glycosidic bond that I just talked about. It's true of all letters, independent of where they are. But there's no force of attraction between letters, similar to the bases. And that allows for any combination to form, any freedom to spell anything, but there's no self-organizational force there. And it shows us that necessity on its own does not explain the specific sequencing and information that we see in DNA. And so if proteins could not encode DNA, or DNA could not form through self-organization by necessity alone, then what? What can? If both chance and necessity alone can't account for what we find in biological organisms, what about both of them working in concert together? And so chance and necessity working together. And this is based off of Darwin's approach, where he explains how new forms can arise as an interplay of chance, through random variations and mutations, and necessity, the law-like properties of natural selection. And could it be that the origin of life actually acts in a similar way? Operin, from the first lecture, he revised his initial theory to account for the increasing complexity seen in proteins and DNA, and he proposed this model. He said if chance alone is too improbable to get fully functioning proteins, what if chance alone just gets you part of the way to a less optimal peptide that kind of partially functions? And then natural selection takes it the rest of the way. It acts on these less than fully functional proteins and then increases their specificity and function to then make metabolism possible and then the cell, thereby circumventing some of the information problem from chance alone. He said the DNA probably also appeared in the same way. But a big issue with this theory lies in this mechanism of natural selection. Because in order for natural selection to occur, what has to happen? You have to be able to reproduce or replicate. That's how you continue to refine things after generation after generation. But to actually replicate is not simple for DNA. This is what it requires. It requires a complex coordination of sequence-specific DNA and protein molecules encoded by the DNA. In other words, it requires information, it requires instruction. And yet to explain the origin of this specified information in DNA, to explain how all this complexity arose, Operin had to invoke a process that actually depends precisely on the very things it's trying to produce. Right? To produce replication, you need DNA. You need the proteins that you're trying to form in the first place. It's like this story from Stephen Meyer. He said a man falls into a 30-foot pit. To get out, he needs a ladder. So he goes home, gets a ladder out of his garage, goes back in the pit, sets up the ladder, and climbs out of the hole. The ladder from the garage, that was his way out. But it begs the question, how did he get out of the pit in the first place to get the ladder? And similarly for natural selection, how did you get the replication to get the natural selection in the first place when you needed those very things like DNA protein to do natural selection? For this reason, Theodore Dobzhansky, one of the leading evolutionary biologists of the 20th century, has insisted that pre-biological natural selection is a contradiction in terms. But still, scientists insist there must be some way in which life can form on its own. As a result, there have been other models that try to combine chance and necessity. I don't have time to go into all of them, but I just want to talk about one of them. And as evolutionary biologists start to see how difficult it was to see how life arose by chance and necessity, they tried to simulate the production of biological information using computer programs. And this was pioneered by Dawkins and Coopers. They attempted to use software to simulate the production of genetic information and demonstrate the efficacy of natural selection. And how they do that? Well, they provided this computer program with a target sequence from Shakespeare. Methinks it is like a weasel. And this is to simulate a desired functional protein. And then the program went through a whole series of steps that I put here, but basically what it did, it generated all these sequences of the same length, 28 characters, made 100 copies, and then mutated all the different sequences. The computer then scored them by comparing them to the goal, and then said which one is the most correct. It preserved the highest scoring string, repeated again, duplicated, mutated again and again and again until it finally got the sequence. And after about 47 cycles, it was able to produce the sequence. This is big news because it showed that natural selection can work to produce a complex, specified sequence randomly. But if you think about it, can you actually see the flaw in the logic here? Well, the biggest flaw that we see here is that nature itself does not have a target sequence in mind. Natural selection only selects based off of function. A sequence is not advantageous just because it gets a sequence a little bit closer to the final sequence. It has to actually function better. Even if an amino acid sequence is closer to the functional protein, unless it has a selective advantage, it would be eliminated. And so looking at this computer simulation, no actual words, functional words, appeared until the 12th round. And this is the word thin in this case. Prior to that, there was no selective advantage, and the only way to pick out one closer sequence to another is to have foresight to what the eventual sequence is going to be. And that foresight is the very thing that natural selection lacks. But of course, which a computer whose intelligence is programmed by human has. And so subtly indicates the need for an intelligence, some sort of intelligent agent to direct the options, exclude others in order to create information. For all these reasons, it's been tough to attribute the origin of life simply to chance and necessity alone, although scientists are still trying to find better theories to explain the appearance of information. And so to do a quick review, where did we go so far? Well, we started by talking about necessity and whether the complexity of life could be explained through chemical forces, like the formation of salt crystals. Dimer experiments showed that amino acids do differentially bind to each other, so it's possible that these properties could then drive the formation of proteins. But then we ran into the issue of whether this theory could explain the origin of information DNA. Either proteins were a template for DNA or DNA self-organized by the same mechanism. The problem of protein template is the fact that there's loss of specificity, there's instability of proteins when they become denatured, and the fact that proteins themselves don't seem to be driven by necessity. Self-organization of DNA, too, is problematic, philosophically because the nature of higher-level information, as well as a need of freedom from restraint in order to convey more information. And then chemically, there are no bonds between the bases in the information-bearing axis. There's no way for there to be differential affinity. And so we then turn to chance plus necessity, where chance takes it part of the way and then refines it through natural selection. But then we ran into the issue of natural selection, where it requires replication, which itself is a complex coordination of DNA and proteins, the very things that we're trying to produce. Genetic algorithms, too, fall prey to issues with natural selection. And so we're still left figuring out the best way to explain the origin of life. You may remember one of the issues that origin of life scientists had to solve was the interdependence of proteins and DNA. You remember when we watched that first video, we saw how interconnected these two things were to produce the materials necessary for life. Transcription, translation, replication all require both DNA and proteins working in concert together. Without either one, the whole process falls apart. So how did they arise? Which came first? And when we think about these questions, we saw the inadequacy of the explanation of a protein-first model. Because proteins are poor templates. To get DNA from proteins reverses the flow of information. We also saw the inadequacy of DNA-first models, as there's a lack of differential binding affinity between bases anywhere along DNA. And the DNA's ability to replicate or actually do anything is dependent upon having proteins. And so scientists are trying to figure out what came first and how. And so they stumbled upon a third way to explain the mystery of life's origins. And they argue that in the earliest stages of life, the environment rose initially from these RNA molecules. And these are molecules which can have both protein-like functions as well as DNA-like functions. And this theory of what we call the RNA world is important to understand because probably the most popular theory today of how life began and establishes a promising framework for how life on Earth could have originated. So to explain this better, I want to play a video produced by Nova about the RNA world.
Where does life come from? This is one of the most important questions humanity has ever posed, and the scientific answer is, we don't entirely know. You might think that cracking DNA's genetic code should have explained life's origins, and it definitely helped. Thanks to our understanding of DNA, we can map out the history of evolution all the way back to single-celled life. But that's where we're stuck. The problem is, DNA is a great way to store information, but it doesn't do much else. Cells rely on other molecules like proteins to replicate, grow, and survive. Proteins, on the other hand, work great as molecular machines to keep cells alive and healthy, but they can't store information or copy themselves. They need DNA for that. So we have a chicken and egg problem. DNA needs proteins to function, and proteins need DNA to exist. So which came first? Which molecule made life possible? Well, there's a third type of molecule that may hold the answer. RNA. Most scientists think that RNA came first because RNA can do two jobs: store information and perform various functions that keep cells alive. This idea that RNA came first is called the RNA world hypothesis. RNA world suggests that billions of years ago, in some primordial soup of molecules, a self-replicating RNA formed. This may have happened in volcanic vents deep on the ocean floor, or perhaps clay clumps brought the necessary chemical building blocks together. Some scientists have even speculated that early RNAs formed on Mars and hitched a ride on an asteroid to our planet. One way or another, self-replicating RNAs emerged, multiplied, and evolved. Over millions of years, they developed into a legion of molecular machines. These microscopic proto-life forms blossomed and competed. The best collections of code lived on, and the weaker ones died out. Survival of the fittest was the name of the game. This competition for survival eventually led RNAs to evolve the ability to build strong, stable proteins, which excelled at carrying out complex biological processes. And somewhere along the line, some critical RNAs mutated into the familiar double helix of DNA. DNA became a stable archive of genetic information that stored blueprints for the most successful RNA and protein molecules. Life became more complex over trillions of tiny steps and happy accidents. And all the while, the RNA lineup grew alongside lengthening genomes of DNA and complex proteins. And it's all still happening inside your body. RNAs have adapted to become the Swiss Army knives of our cells. Today, they can slice, dice, catalyze, build, destroy, code, replicate, and transform. A remarkable diversity from the simplest of beginnings, a single self-replicating RNA molecule.
Man, that sounds pretty good. RNA seems pretty awesome. You're able to do both the function that cells need as well as store information and replicate in order for there to now be natural selection. It addresses many of the issues that we've been bringing up. And then over time, as the chemical evolution progresses, gives way to proteins and DNA so that we have the system that we have today. QED, end of story. But actually, as you look closer to the details, there are actually a bunch of issues that we need to account for. And there are many reasons, but there are three that I want to go over today. I don't have time to go too in depth on each one, so if you're really interested, read Meyer's book, Signature of the Cell, chapter 14, upon which this talk is based. Okay, but there are three reasons why the RNA world really doesn't hold water. First of all, RNA building blocks are hard to synthesize and easy to destroy. The building blocks of RNA are the bases adenine, cytosine, guanine, and uracil. It also has phosphate molecules and a sugar called ribose, very similar in structure to DNA. But forming these and maintaining these in the prebiotic soup is actually proven to be very difficult or near impossible. First of all, nucleotides are actually hard to synthesize. In the origin of life experiments, these nucleotides actually were never ever formed. And then what we find is that they're also very unstable in the early environmental temperatures. At the time, they were really, really hot. And we find that at even 100 degrees Celsius, cytosine, one of the bases, has a half-life of only 19 days. That's extremely short compared with the time necessary to form the first functional RNA enzyme by trial and error. And scientists have also found that also producing the sugar ribose is problematic as well. Ribose is thought to arise from something called the Formose reaction. Starting with formaldehyde and water, you get a whole bunch of different sugars, including ribose. But all this gets inhibited in the presence of nitrogen-containing molecules, of which there's amino acids and nucleotide bases. And yet these very elements are what's required for the formation of RNA. And so looking at all these things together, Dean Kenyon said, the chemical conditions predisposed for prebiotic synthesis of purines and pyrimidines are sharply incompatible with those proposed for the synthesis of ribose. Right? Again, the conditions for needing to produce these bases are very, very different and actually opposed to the formation of the sugar itself. So I think that's number one. Number two, ribozymes are poor substitutes for proteins. Naturally occurring ribozymes are actually very few of the enzyme functions of proteins. They can only do these three things. They can cleave another RNA by hydrolysis. They can ligate or stick together separate strands of RNA. And they can promote peptide bond formation when in a ribosome. That's only a small subset of the thousands and thousands of functions a cell needs. It's like saying, I only need a hammer to build a house because it performs some of the things I need. No, life in a cell requires a lot more than that. And then thirdly, the RNA world doesn't explain the origin of genetic information. The problem still remains. Where did all this information in RNA come from? Notice in the video they couldn't really explain it. Volcanic vents, clay clumps, Mars? It's actually the same problem that we run into as DNA. We can't appeal to chance because the information necessary to build just one protein of modest length is astronomical, let alone all the proteins necessary for life. Neither can we appeal to self-organization, because just like DNA, there are no binding affinities that can account for the specific sequence of bases. Some scientists have tried to point to RNA molecules and say, well, perhaps they arose by chance, that over time they kind of got partially functional, and those that carry enough complexity to self-replicate, these then get selected for and reproduce. But even then, when you think about it, the capacity to self-replicate is not easy. Most RNA molecules actually have no ability to self-replicate. Scientists, they've tried to engineer self-replicating RNA molecules. Even when they try to engineer the right molecules, still less than 1 in 10 to the 15th are able to theoretically, to even partially self-replicate. If you do it by chance, it's even much more rare. And in order to self-replicate, they've also found that these RNA enzymes need to be about 250 bases long in order to have the proper template binding as well as form the 3D shape necessary for replication. That's 1 in 10 to the 120th possibilities. I mean, it's astronomical to form by chance. It's highly, highly unlikely. And so Robert Shapiro, a chemist, he analogized all this property of self-replicating RNA molecule out of chance to a pile of Scrabble letters. And he says, when you fling them onto the lawn, what's the probability they'll form the words to be or not to be? That is the question. He said, that is similar to the odds of an RNA molecule randomly forming into a functional RNA that could copy itself. And so these things are problems that the RNA world has to deal with. And so when we think about that, where are we now? Well, we saw that chance, necessity, and the combination of chance and necessity, they're not enough to explain the origin of information that we see in DNA and proteins. In addition, when we frame the possibilities different, things don't change. The protein first theory failed, so we went to DNA first. That failed too, and it gave rise to RNA world, which still fails to explain the central issue of the origin of information. So could it be that there's another explanation? One that information has been pointing to the entire time, the idea of an intelligence. When we think about our life experience, we see that large amounts of specific information, they tend to not arise spontaneously. In fact, almost always they arrive from intelligent sources. In fact, always, I would say. For example, there was that flower example I used back in the first lecture. Imagine you saw a pattern of flowers that had a nice design and they spelled out "see you next year." You don't say, wow, look at that amazing pattern that the force of nature happened to make. You instinctively know it's not a random event. Even if you didn't see the flowers planted, you didn't see them arranged, you know looking at that there must have been some sort of intelligence, probably a gardener, behind it, even if you didn't see it. Now, when some people think about this word intelligence, they think, well, that's not scientific. That's a cop-out answer. You're saying that just because you can't explain things. No, that's not true. In fact, scientists from many fields recognize the connection between intelligence and information. And for them, intelligence is a perfectly rational explanation for things that they observe. For example, when archaeologists went and they discovered the Rosetta Stone, they assumed a scribe produced inscriptions rather than it being a random series of markings just by looking at it. Evolutionary anthropologists, they established intelligence of the early hominids from chipped flints that was too improbably specified in form and function to have been produced by natural causes. And then, of course, SETI, the example I used before, specified complex information indicates some sort of intelligence out there. And so similarly, when you look at the specified complex arrangement of nucleotide sequences, the information present in DNA, it seems to imply the past action and existence of an intelligent cause, even if it cannot be observed. But the question is, can this intuition that we have, can that be quantified? Well, William Dembski, a mathematician, he developed a scientific method for determining and detecting designed events. In his book, The Design Inference, he identified that intelligent activity has the joint properties of these two things that we've been talking about all throughout this talk. Ones of complexity, whether it's improbable and exhibits an irregular arrangement, and specificity, specification. Does it correspond to an observed event or a functional pattern? And when we observe these things, we instinctively start to compare the competing theories of patterns, of necessity, or intelligence based on the probabilities and signatures of the events that we see. And in fact, what we do instinctively is we put on some sort of explanatory filter to the events that are before us, determine is it by necessity, by chance, or design. And Dembski presents it in this schematic, which he titles the explanatory filter. And he suggests this, that we commonly attribute high probability events to natural laws or necessity. Events of intermediate or low probability we ascribe to chance. And we attribute specified events of small probability to intelligent design. And so let's go through this with a few examples to see how this filter works. Going back to a roulette wheel. What if we were testing a brand new wheel and we spun it 100 times and this is what we got? First we got 16, and second time we got 16. Wow, that's good, right? That seems improbable, but by chance, yeah. But then say you start spinning again and again. You start to get 16s 100 times in a row. At this point, you're like, wait a second, something else is going on. Why? Because a seemingly low probability event has all of a sudden become high probability. There's a repeating pattern again and again and again. There must be something underlying underneath it. And what we're sensing is what we see here. That high probability event points to some sort of necessity or physical law. And sure enough, if you were to inspect that new wheel, you'll notice a high lip on the 16 pocket, causing it to fall in the same pocket each time. But what if I go back to the casino? Now a normal roulette wheel, it's functioning normally. The first trial I bet on 16. Maybe out of 100 tries, I get 16 maybe twice. That's a low probability, 1 over 38 to 100th power. There's no pattern. And so you would look at that and say, wow, you won it by chance. The wheel's not rigged. But now I say I go on the same wheel again. It's not defective. And I bet 16 every single time. And the second trial, I get 16 every single time, 100 times in a row. It's exactly the same probability as the first case, but what's the difference? Having all these 16s conforms to a specified pattern that I had, quote, called, which I had bet on. It wasn't out of necessity, and so it makes you think that there's something very fishy, some sort of design that's too good to be true, low probability with a specified pattern. And we can take the same inference into nature. Take, for example, when visitors go to Mount Rushmore in South Dakota. When they look at it, immediately they infer the action of intelligent design, looking at all these unusual shapes etched in the rock. And why? Well, the shapes themselves, they're unusual. They're irregularly shaped, and so they are improbable. They're not out of necessity. But then why not chance? Why not when the wind blows, the rains come? Why can't that create something like this? It's because they see a pattern that they can recognize from pictures of past presidents. It matches a pattern that they have observed elsewhere, and so exhibits specification. It points to some sort of intelligent design. Same with language. When we see the words on a page that convey ideas, the arrangement of letters, that's too complex to just be necessity. And yet, it carries meaning, has a specific pattern that eliminates chance. And so it infers some sort of intelligence, a writer who wrote it. And so now, when applying this filter to DNA, what do we find? It's obvious that the base sequences of DNA are improbable. They're complex, or it's not out of necessity. But do these base sequences match a pattern. Are they specified? And we don't necessarily see a pattern visually, but there is a pattern functionally. These sequences, they correspond to a functionally significant outcome, the synthesis of a protein. And so when we look at the spectrums of the possibilities for the requisite amount of life and biological information, the specified complexity that we see in DNA and proteins infers that intelligence actually seems to be the best explanation. Of course, this can't be proven without a shadow of a doubt. But I do think these reasonings, as you walk through the different possibilities, they provide strong evidence that there must have been some sort of intelligence at work to give us the information that we're seeing in DNA and proteins and then give rise to life. As a quick review of what we went over so far in the second half of the lecture, what did we do? Well, we looked first at the RNA world hypothesis, how it seemed to combine the information-bearing properties of DNA with protein-like functions. But we saw that there are issues with it, that the building blocks are hard to synthesize, easy to destroy. Ribozymes only cover some of the essential functions of proteins. This hypothesis doesn't explain the origin of information. So with these different approaches having issues, we looked at intelligence. We saw that in our everyday experience, when we see large amounts of specified information, we attribute intelligence. And it's not just us. We do this in different sciences as well. And so then we discussed ways in which we can determine intelligence, which was quantified in Dembski's explanatory filter. We looked at different examples of how to use it with roulette, Mount Rushmore, and then language. And then using his filter, we then looked at the features present in DNA, its complexity, specificity, and we found that perhaps intelligence is the best explanation for the origin of life and its biological information. And so, what we found is that is true, that perhaps intelligence is the best way to explain the origins of life. So that's it for our origin of life section. It's a lot of stuff. So take some time now to review and consolidate what you've heard and what you've learned. So we'll see you next week.